Hello, and welcome to the third section of our course. My name is Stella Aslebekian. I'm an epidemiologist who works with genetic and epigenetic data in the context of chronic disease. In this section, we will learn how to apply Mendelian randomization techniques that you've already learned to causal inference in the epigenetic setting. But before we do that, I would like to offer you a bird's eye view of the field of epigenetics in general and to show you its promise and its challenges. Today, we'll start with a brief history of epigenetics and what exactly it studies. We will cover the major types of epigenetic changes, focusing on DNA methylation, which is the best studied class of those. We will talk about how we measure and quantify epigenetic processes and discuss whether or not these markers are heritable, which is actually an ongoing controversy in the field. Finally, I will offer some thoughts on the applications of epigenetic technology in large-scale epidemiologic studies. Because we have so much ground to cover, this lecture will be a little bit longer than the rest of the lectures in this module. So let's begin with the basics. You can see from these journal covers and magazine covers from the past decade that epigenetics has emerged as a very hot area of scientific discovery. There's a lot of enthusiasm about epigenetics because it's believed to have this potential to modify your DNA inheritance and thus affect health, hopefully in a positive way. You can see here that the Time magazine declares your DNA is not your destiny, and Der Spiegel, which is the German magazine next to it, goes even further and claims victory over the gene. That sounds very exciting, right? But what is epigenetics and why are we so interested in its promises? The most common definition is right here on your slide. It is a branch of science that studies heritable alterations in gene expression that are distinct from actual mutations in the DNA sequence. You will notice this word mitotically. For those of us like me who may be a little rusty in cell biology, that means that an epigenetic marker can be transmitted to future generations of cells through simple division or mitosis. So we start with the parent cell has an epigenetic mark, the daughter cells would also have the same epigenetic mark. That applies to cells, not whole organisms. It does not mean that those epigenetic marks are also necessarily inherited across generations of organisms because that would require meiosis. We will talk more about inheritance later today, but for now, keep in mind that epigenetic markers absolutely can be transmitted during simple cell division. That's an important nuance to remember. A quick historical detour here. The word epigenetics is actually not new, even though this enthusiasm is. The word itself, which in Greek means above the genome or beyond the genome, was proposed by a British developmental biologist named Conrad Waddington in the 1950s. Waddington was a geneticist and an embryologist who studied fruit fly development, and he was interested in reconciling the nature and nurture question. And he created this canonical diagram of embryonic development. Where the ball starts, that's the moment of conception, and that's determined by the genome sequence itself. But where the ball ends up over time, phenotypically speaking, is determined by various combinations of the starting point, which is the genome, and the environmental landscape. So even though Waddington, like a lot of early geneticists, did his work in fruit flies, the same concept, of course, holds true in other organisms, and that includes humans. Here's a more modern analogy, and that's a computer system. Here, the genome represents the hardware, which is unchanging, while the epigenome can be viewed as the operating system of our computer. There are signaling cascades that transfer information between the outside and the inside of cells, and that's your software. Your hardware doesn't change, that's your genome, but both the software and the operating system, the epigenome and the phenome, are malleable, and they could potentially be updated or edited, even though, of course, especially in the human setting, that's a lot easier said than done. So what determines how or if your operating system will be updated? And it's really two things. The first one is the DNA sequence itself. Which nucleotides you have in a given region can determine whether or not that particular point in the genome is likely to be epigenetically affected. For example, In the DNA methylation setting, which is the example right here in your slide, we know that most methylation occurs where a C nucleotide is next to a G nucleotide. So if you have an A or a T there, it's less likely to be methylated. That's the hardware contribution. The second piece of the puzzle is the environment, and that's defined in the broadest way that you can imagine. Some examples are right here on the slide. Such epigenetic modifications can occur throughout an individual's lifetime, but at certain periods, for example during fetal development, which was Waddington's interest, epigenetic activity can be higher than during other periods. The take-home message here is that both the genome and the environment come together to determine whether a certain genomic region is likely to be epigenetically affected, in this case methylated. And these epigenetic modifications can then go on and up or down regulate gene expression and influence the risk of disease. Here, I wanted to really hone in on the importance of the environment. In this module, you will also listen to a TED Talk about the importance of early life care as a critical genetic, or rather epigenetic environmental factor, although he does talk about genetics a little bit, because again, they're all connected. So here are some other examples of environmental factors that have been shown to be associated with epigenetic changes in humans. As you can see here, clockwise, we have things like diet, drugs, and that can be either prescription drugs or illicit drugs. Certainly we know smoking has been conclusively linked to epigenetics, physical activity, financial status, social connections, adverse experiences like discrimination. So it's really a wide range of environmental factors that are not just lifestyle, that can modulate gene expression, that can become embodied, epigenetically speaking, and can turn genes on or off, up or down. So now we have defined epigenetics as changes in gene expression that can arise due to both genetic and environmental influences. Let's take a closer look at the biochemistry of epigenetics. This figure shows three basic groups of epigenetic modifications. I already mentioned DNA methylation. It is exactly what it sounds like. It's a methyl molecule, CH3, attaching itself to the DNA double helix. Some other types of biochemical modifications can be post-translational, which means they affect proteins rather than DNA. For example, histones, which are proteins, can undergo phosphorylation, methylation, and acetylation, among other chemical modifications. These modifications are involved in chromatin remodeling and also affect transcriptional regulation and gene expression. Finally, since we're on the subject of transcription, the RNA molecule can also be epigenetically modified, just like the DNA molecule or histone proteins. Normally, mRNA would be translated into protein, but that process can be affected by binding of microRNAs, methylation, or other epigenetic changes. You might note here that this is a slightly older figure, and while it's still true as far as the basic classes of epigenetic processes, we know now that heritable silencing, which is here in the middle, is not the only effect of epigenetic processes. Genes are not only silenced by epigenetics, they can be turned up or louder too. It all depends on where in the genome these changes are and what circumstances may be that govern those processes. And by circumstances, I really mean environment. Also note that there are arrows connecting these mechanisms, and that's a pretty accurate representation of the biological complexity of epigenetics. Not only do we have to think about all these different classes of epigenetic modifications, we also have to think about how they interact. So let me say a few more words on the major classes of epigenetic modifications. This slide shows you the variety of different biochemical actions that can befall histones. I already said that these modifications are involved in chromatin remodeling and transcription, but in plain English, that means those modifications establish how accessible the DNA is to transcription, and thus that can regulate gene expression and affect phenotype. Note that here too, epigenetic modification does not always mean repression of transcription, but can rather up or down regulate gene expression depending on circumstances. Here are also some examples of the RNA-based mechanisms on this slide. This is by far the least understood class of epigenetic mechanisms, And every once in a while, we discover new ways that RNA can be modified. We have non coding RNAs, RNA interference, microRNAs, RNA methylation, and so on. All those processes tend to affect the mRNA's ability to be translated into proteins, and thus their ability to influence the downstream trait. I've saved the best for last, or at least the best studied for last, and that's DNA methylation. DNA molecules can be methylated by the addition of a methyl group, which is right here, that pink circle with CH3 in it, and that's added to carbon position 5 on cytosine bases. That chemical reaction is catalyzed by DNA methyltransferase enzymes, and those can help maintain the resulting changes in gene expression. Again, here the slide mentions silencing, but that's canonically the case if the methylation is in the promoter region of the gene. As I said before, DNA methylation may actually upregulate expression, especially if it's located within gene bodies. We're back here to our old figure. Perhaps I should call it a classic figure. It's an oldie but goodie that faithfully summarizes the basic classes of epigenetic modifications. For the rest of this course, we will focus on DNA methylation because we have the technology to interrogate it in a cost-effective way, and that means we can use it in large-scale epidemiologic studies. So, how do we interrogate DNA methylation? Let's spend a few minutes on that. To me, it's the quintessential example of cheap technology driving scientific discovery rather than vice versa. We do it because we can. So how can we? I mentioned before that about 80% of all DNA methylation occurs at so-called CpG sites. That's just shorthand for cytosine phosphate guanine region of the genome, or CG dinucleotide. It's a place in the genome where C is next to a G. CpG islands, which are just full of those CG dinucleotides, often occur near promoters of genes, and this is the classic paradigm of CpG methylation and how it affects downstream gene expression that's shown here. We now have commercial arrays that can interrogate hundreds of thousands of CpG sites, including but not limited to those that are located in such CpG-rich islands. These arrays are made by Illumina in California, and they cost about a few $100s per sample. This is 2018, and the last quote I got was about $350 per sample. If you wanted the really gold standard, though, you could also do bisulfite sequencing to measure methylation. But that would run you, as of now, over $1,000 per sample, so it is rarely done. In our group, we have shown that the concordance between the two methods is quite high, so you might as well go for the cheaper array. Those arrays output a methylation statistic at each CpG site. That's called the beta score. And please do not confuse it with the beta regression coefficient. The epigenetic beta score is simply the ratio of the methylated probe intensity and the overall intensity of the signal that's generated by the array. In plain English, it's kind of like a methylation proportion. Sometimes you'll see something called M values, and that's essentially a logit transformation of the beta score. M values behave a little bit better mathematically, but beta scores are easier to interpret because they're just that methylation proportion, and they're more commonly used because of that. On this slide, I have listed the available Illumina methylation arrays in their order of appearance. The most cutting-edge one, as of 2018, is the EPIC array, and it interrogates about 800,000 CpGs located across the genome. The diagram here shows the distribution of DNA methylation in specific gene regions. The overall distribution and the mean beta value of the CpGs in each region are here shown as box plots. You'll see that the least methylation is observed in promoter regions and the first exon, while the most methylated regions are in the gene body and 3' UTR. The numbers on the x-axis here correspond to the total number of CpGs in any given gene region on the 450 chip, which is not the most cutting-edge one, but that's the one that's really widely used, so you might as well know where in the genome you're looking. Okay, no more biochemistry. Let's talk about this fun question of inheritance. And before we go any further, I want to ask you a question. If you wanted to test whether epigenetic marks such as DNA methylation are transmitted across generations of people, how many generations would you need? And let me throw you another curveball. Would that number of generations be different for maternal inheritance versus paternal inheritance? Think about that. How many generations would you need to test epigenetic inheritance? Pause the slide if you need to, and then come back. When people think of epigenetic inheritance, they usually imagine something like this. The environment and or the DNA sequence confer certain epigenetic modifications. Those modifications affect gene expression, and ultimately they affect translation into proteins. The parent acquires a certain phenotype through epigenetics, and then that parent passes that phenotype on to the offspring through those epigenetic marks. It sounds really Lamarckian, right? The giraffe's neck extended so long because he needed to grab leaves off the top of the tree, and that subsequent generation of giraffes is born with their neck already extended. We know now that's not how evolution usually works. But perhaps Lamarck was right on a micro level. Perhaps there are exceptions, and the exceptions would be such epigenetic inheritance. Is there such a thing? To answer that question, I want to go back to the question I asked you earlier. To test that hypothesis, how many generations do you need to observe? Are you ready for the answer? The answer is 4 on the maternal side and 3 on the paternal side. Let's go through this together. Let's say we have a pregnant woman who is smoking, and there's a lot of very good evidence that links smoking to epigenetic changes. She's exposing herself, right? That's the first generation. She's exposing her fetus, that's the second generation. And in this case, her fetus happens to be a little girl. So she already has all her eggs. And those eggs are also getting exposed. Those eggs will make up 1/2 of the grandchild. You can see these generations here, they're color-coded. And you can trace the granddaughter back to the oocytes that are currently being exposed to smoking. So if you see the same epigenetic change in the grandchild as you see in the grandma, it does not have to be inheritance. It can simply be because that granddaughter, as an egg, was exposed to smoking. So whatever grandma was exposed to, the daughter was exposed to, and the granddaughter was also exposed to. So to see if you can inherit the changes, you would need one more generation, and that makes 4. In case of sperm and the paternal inheritance, you need 1 generation less because the sperm are not formed in utero, but are rather formed throughout the life course. So the logical question is, have studies looked at that? Have studies looked at 4 generations to ascertain epigenetic inheritance? Some have, especially in animal models, and I'll tell you right now that the evidence for that kind of true inheritance is very scant. Here's why. At conception, we undergo not 1, but 2 cycles of demethylation to erase all the epigenetic marks accumulated by the prior generations. There is a theory that it confers an evolutionary advantage because the epigenetic marks on the germ cells of the parents were confirmed by one environment, but the fetus may be born in a different environment, so it behooved the fetus to adapt, epigenetically speaking, not to the past but to the future conditions. Here you can see that the levels of DNA methylation plummet twice throughout the very early embryonic development. So even though epigenetic inheritance may exist, it is very, very hard to find because of this phenomenon. In the final part of this introductory lecture, we will talk about the good, the bad, and the ugly of using epigenetic data. And again, mostly that's DNA methylation data. And we'll talk about how we use it in epidemiology. Before we got our hands on epigenetic data, and by we I mean epidemiologists, there were compelling reasons to believe, from animal studies and other models, that epigenetics played a really important role in disease risk and was worth investigating. Particularly, we were inspired by a lot of studies in oncology where tumor epigenetics was successfully leveraged to target treatment. As of now, the FDA has 7 epigenetic drugs approved for cancer treatment. So that was inspiring. On the more research side of things, we noticed that epigenetic markers explain a much higher percentage of trait variability compared to a single DNA sequence mutation. One CpG locus in one of our studies has been shown to explain as much as 5% of lipid trait variation when all the known mutations could explain only about 8%. So one CpG versus many, many SNPs, and the estimate of variance explained is comparable. Then we have this issue of technology, right? We can interrogate epigenetics cheaply, we can do it efficiently, and really, for me personally, I like it because it's a really elegant way to connect the genes and the environment in a way that makes biological sense. So why not? But here's the thing. Epigenetics is tricky. It is much trickier from an epidemiologic causal perspective than garden variety genetic epidemiology. Unlike the genome, the epigenome is not static. Your epigenetic marks are not just established once during conception like sequence mutations. They're accumulated throughout your lifetime and can even be reversible. Here on this slide, you see genes and rather epigenomes from identical twins. They have the same DNA, and at 3 years of age, they have very similar epigenomes too. Have you ever noticed that newborn twins look really, really alike, so we make all those jokes about switching them? Well, if you look at those same twins a few decades later, they look more like siblings. And not just because they're wearing different clothes. It's also because of their divergent epigenomes that reflect different environmental exposures of these twins. And that's what you can see here in the second panel. So time matters. This is something we need to account for in any epidemiologic study involving epigenetics, is that time course of epigenetic changes. The other kind of specificity that makes this really difficult is tissue specificity. All your cells have the same DNA blueprint, but your cells look very different, right? Your neuron looks kind of like a tree, and your adipocyte looks kind of like a blob. Why is that if they have the same DNA? Well, because they have different patterns of gene expression, and that's because they have different epigenomes. In epidemiologic studies, we usually have access to blood or saliva, but sometimes we may want to think about changes in the heart or the liver or the brain. So if different cells have different epigenomes, does it make sense to use blood or saliva as a proxy for the liver or the heart? And panel B here is not especially encouraging in that respect. At best, you have about a 20% correlation. I circled the CD4 positive lymphocytes, which is what we use in our studies. They do not do particularly well. So this is something you also have to think about, is how well are you measuring what you're trying to measure, which is the epigenome and biologically relevant tissue. And on the left here, you can also see that the correlation and methylation also really depends on how far the CpGs are from each other. So there's a lot of complexity here to consider. So you have the issue of temporality, because you really cannot know if the epigenetic changes came before or after disease onset, and tissue specificity, because you do not know if the data you're getting from blood or saliva actually reflect the underlying biology. And the next challenge is reversibility. If epigenetic markers can go away, well, when should we measure them? And this diagram shows that this reversibility can strike at any point. We have the central dogma of molecular biology on the left. You go from DNA to RNA to protein. At each of these levels, we can have epigenetic modifications, shown here on the right. And at each of these levels, these modifications can be reversed. That throws another wrench in the timing of your measurements. Finally, that very same aspect that I like about epigenetics, the synthesis of genes and environment, makes it tricky because it generates an inordinate amount of data. The environment is also very hard to measure. It can change, it can be situation specific, and because of all these complexities, it is much harder to establish causality when it comes to epigenetics compared to simple studies of DNA mutations like GWAS. So as we wrap up this introduction to epigenetics, here are some take-home messages. Epigenetics is very exciting. It's the new frontier in personalized medicine, It's already proven itself in oncology, and it answers several nature or nurture questions. And of course, the answer is both. But epigenetics is also a double edged sword. What makes epigenetic markers such good targets for intervention also makes them an absolute nightmare from the standpoint of causality. So, in the next part of the module, we will talk about how we can use Mendelian randomization to infer cause and effect from epigenetic data.